Hello. Today's lesson is on species and taxonomy. I'm going to try and keep it a bit shorter today so that you can go through most of the work yourself in chapter 10.1. Okay, so what are we going to be finding out about? We are going to be finding out about what species are. You should remember from GCSE that a species, the definition of a species is when two organisms mate, they are able to produce fertile offspring. So we know that some organisms can mate with one another and produce infertile offspring. You could think of the liger or the tigon, which is a tiger and a lion mating. They do have offspring, but those offspring are infertile. So that's showing that they are no longer part of the same species. This definition is slightly more flexible these days now that people know a bit more about relationships between different species of animals. But for our purposes, this is what we're sticking to. Okay, so when we talk about species, we're aware that there are lots of different types of living organisms, lots of different species of organisms. So we're going to talk about how we actually classify animals as a specific species. So you might know the scientific names of some of these organisms. If we look at them, Homo sapiens, that's ours. A prunus sp, so that's saying that's the genus Prunus, and we'll talk about that in a minute. And it's a particular species, so you get lots of different kinds of prunus plants. So you get plums and peaches and apricots. So you could get a family of them. So we're saying this is definitely a prunus one. Might not know exactly which one it is. Lupus lupus, the wolf, and Canis lupus, the dog. And you can see I've written these names in italics. It's important when we use this binomial naming system, binomial, two names, that we write it in italics. If you can't write it in italics, because most of us don't actually write in italics, so if you can't do that, then you would simply underline the two names to indicate that that is the binomial name. So if you're writing an essay and you wanted to write lupus lupus, you would simply underline them. The genus name would have a capital letter, so that's the first name, and the second name, the species name, would have a small letter. Okay, and that's the binomial system. Binomial system, you might remember some of this from GCSE. Obviously, you might remember was discovered by a man called Carl Linnaeus, and we did that again, sorry. And Carl Linnaeus was a very particular man. He had an eye for detail. He was really into classifying things. He went around looking at the details of grasses and eventually came up with this massive classification system that we still use today. So here's a short video about Carl Linnaeus. So if you want to go away and watch that, please feel free. It's very short and just to give you a little bit of a history of the classification system and how he developed it. All right, so the way we classify things, we use something called phylogenetic classification. So phylogenetic classification is where we classify organisms into hierarchies. And within those hierarchies, we put them in groups, and those groups are non-overlapping. So an organism cannot be in more than one group. The highest hierarchy that we can have is a domain, and I'll talk to you about what the domains are. Below the domain, we have kingdom. We have the kingdoms of life, so we fall into the animal kingdom. And then below that, we have phyla, then class, order, family, genus, and species. When I spoke about the binomial name earlier on, the two names that are being given are the genus name and the species name. So for us, Homo sapiens. For the wolf, lupus, lupus. For the dog, Canis lupus. Okay, so the last two, genus and species, they give the binomial name. These groups are called taxa. So each one of these is a taxon. So taxa, plural, taxon, singular. Okay. So let's look at a specific example here. Let's look at the fox. So the fox is found in the domain of eukaryotes. So we know what eukaryotes are because we've compared eukaryotes to prokaryotes. Eukaryotes are a domain. There are 3 domains of life. I'll talk about that in the next slide. Within the eukaryotes, the fox is an animal. So it finds itself in the animal kingdom. It could, there is the plant kingdom, there are fungi, there are the protoctista or protists as well in the eukaryotes. But the fox, it finds itself in the animal kingdom. Within the animal kingdom, it finds itself in the phylum Chordata. Chordata means it's got a cord, it's got a spinal column. Within that Chordata classification, within that taxon, it then finds itself a mammal as opposed to a reptile or a fish, which are also animals with backbones. Within that mammalian class, it finds itself in the order of carnivore. So it's a carnivorous animal. Within that, it finds itself in the Canidae family, which is going to be the family of dog-like animals. Then Vulpes is the genus, and then species, genus, species, Vulpes, Vulpes. Okay, so this is the common hierarchy that we use, the common hierarchy of taxa, and you obviously need to know the order of these. So let's look at how they establish that hierarchy. They establish the hierarchy using phylogenetic trees. Now, phylogenetic trees have classically been based on evolutionary relationships. So what would have happened is scientists would have looked at different organisms and they would have found what was similar between them in terms of how they look. So what characteristics they actually share. And it would be quite complex, looking at their skeleton and looking at maybe homologous features such as the fin of a fish compared to the arm of a human compared to the wing of a bird, for example. So you have these homologous structures that you can compare, and you can say, well, at some point in time, possibly they were related in evolutionary history. These days, however, we're more inclined to use a more precise method of comparing sequences, either sequences of DNA or sequences of protein. So this is what a phylogenetic tree might look like for a fox. So here's our red fox up here. And what this phylogenetic tree shows us, so if this is red fox over here, what this shows us is that the red fox shares a common ancestor with something called a Ruppel's fox. And the Ruppel's fox and the red fox are then next most related to something called a Corsac fox. So they all share a common ancestor. If we go further back in time, we can see that they are closely related to the Arctic fox and the kit fox. This also suggests to us that the red fox is not very closely related at all to, for example, the dog. The dog and the red fox share a common ancestor. Let's have a look where their common ancestor is. They share a common ancestor here. So this is where the speciation happened to form 2 different species, and it's where the fox and the dog would have diverged. So we're looking, so these boxes here are telling us about time, telling us that over 10 million years ago, the dog and the red fox would have shared a common ancestor. So these phylogenetic trees are really, really helpful. And they are, like I said, determined by comparing DNA or protein sequences from the animals. So what they might do is they might select an enzyme that's common to all of the animals and compare the sequences of those enzymes. If those enzymes are very similar, you could say that these animals are very closely related. If they are very different to one another, you could say they're less closely related. So here is an example over here of protein sequences being compared. And you can see that there are places where there is some homology. So where there is red, there is homology. That means that they are the same. And where they are black, they are completely different. And where they are blue, this is telling us that it's a different amino acid, but it's got a similar type of chemistry. So there is a very, very low homology here, very low homology. So there's not a lot of similarity between these 2 proteins. So this is one protein and this is the other. So we'd say that there is not that these animals would not necessarily be very strongly related. If we had to compare a DNA sequence, here we've got two DNA sequences that are being compared together, sequence number 1, sequence number 2, where you can see the lines, that's where the sequence is the same, and where there's a gap, that's where the sequence is different. So if we have to take the whole sequence, we can say at the end of it, they're 82% similar. So they might be related at some point. They're probably not very, very closely related. They're probably not the closest, they probably don't share a common ancestor in recent history, but there is clearly some relatedness. So that's how we establish these phylogenetic trees. Okay, and if we look down here, we can see we've got our red fox, we've got a dog, we've got grey wolves over here, African wild dogs, and then even further back in time. So that's where, you know, there's a branch to black bears, pandas, walruses. So obviously a long time, you know, many millions of years ago, they would have shared a common ancestor there. Okay. So I said I'd mention the domains. The 3 domains are, bacteria, archaea, and eukaryotes. So here we've got bacteria, archaea, and eukaryotes. So this is drawing it in the style of a phylogenetic tree. We can see our eukaryotes are broken into a number of kingdoms. Our eukaryotes make up our animals, our fungi, plants, and the protists. Plants are photosynthetic, and some protists are photosynthetic. You might know algae are not classified as plants; they're actually classified as protists. So they can be single-celled, they can be multicellular, and they are photosynthetic, but they are not plants. Then, in a totally separate domain, we have the archaea, which are a specific type of unicellular organism, which used to be grouped together with the prokaryotes, but they realized actually they had quite a few similarities with the eukaryotes, and they had enough difference to stand alone. And then our bacteria are prokaryotes, which we have studied. So these are our typical bacteria. In our archaea, we are likely to find our extremophiles, so the bacteria that live in really hot conditions or in sulfurous conditions or in very salty conditions. Those are our archaea microorganisms. So we can see here in terms of a phylogenetic tree, if this is sort of the beginning of life, how it's branched out, and that protistas all were very, very early, then our fungi, and then later on our animals and plants would have branched off there. Okay, so you need to be aware of the 3 different domains, and you do need to be able to compare the differences between bacteria and archaea and eukaryotes. So have a look in your textbook, summarize that, make some good notes on that. Okay.